Hello. This is a tutorial about the SbmlComp, and how to interpret the tables and graphs in its analysis report. To follow this tutorial better, we recommend watching it in high quality. To change the video quality, you can go to Settings and select 720p or 1080p. This is the analysis report of a single model. It is divided into sections and subsections. By clicking on this blue bar, you can collapse or expand the sessions. As you can see, this web page has two sections, Model Analysis and Export Models. If two or more models are used as input, another section with the model comparison results would also be present. The Model Analysis section starts with the subsection General Characteristics, showing several aspects of the model and information used on the subsequent analysis. First, it is shown the selected name of the modeled organism on the KEGG and BioPsych databases. In this particular example, no organism name was selected, so the KEGG organism is shown as entire database, and the BioPsych organism is shown as MetaPsych, Tier 1. The MetaPsych, Tier 1, corresponds to a fraction of BioPsych, and contains only curated pathways. Then, you find information on the number of compartments present in the model. In this example, the model has two compartments. You can also find the number of metabolites and how many of them have synonymous in multiple databases, including KEGG and BioPsych. The reactions present in the model are classified into 5 categories. Metabolic reactions, which are balanced reactions that do not transport metabolites between compartments. Exchange and sink reactions, which play a role similar to that of the growth medium. Transport reactions, which transport reactions between compartments in the model. Demand reactions, which are helpful to prevent metabolites from accumulating inside the cell. And inactivated reactions, which cannot carry flux because their minimum and maximum allowed fluxes were set to 0. In this example, E. coli core has a total of 95 reactions, among which 50 are metabolic reactions. Then, you find information about the number of blocked metabolic reactions. The blocked reactions are those that cannot carry flux, but were not intentionally inactivated. They depend on the growth medium, since a model without a medium should not have flux on any reaction. For this analysis, the culture medium provided by the model is enriched with all the metabolites present in the biolog phenotype microarray experiments, and only the reactions that still cannot have flux are listed here. Below, you find information on the number of synonyms for metabolic reactions in various databases. The number of metabolic reactions in the previous table corresponds to the total in this table. In the second subsection, the KEGG pathway modules, the model's metabolic reactions are mapped to KEGG reactions and KEGG pathways. To map the reactions, we consolidate translations of metabolites and reactions from the model file itself, and the MNX-ref. It is important to note that it is often not possible to map all reactions in the model to KEGG and BioPsych identifiers. To have a better estimation of the number of translated reactions, you can check the last table. For this model, 38 reactions were mapped to BioPsych, and 46 were mapped to KEGG. Let us use an example to explain better how the mapping of reactions is performed. Let us say that your model uses big identifiers, and you want to know if the reaction FUM is synonymous with the KEGG reaction R01082. For the two reactions to be considered synonymous, the metabolites of the two reactions must also be synonymous. The metabolites are considered synonymous if this information is present in MNX-ref, or in the model file. Checking the MNX-ref database, FUM is synonymous with C00122, H2O is synonymous with C00001, and mal__L is synonymous with C00149. For the two reactions to be considered synonymous, they should also have identical stoichiometry. Additionally, the model's reaction directionality should also be checked. In the model, it can be bidirectional, forward, or reverse. If the reaction in the model is not bidirectional, we look for pathways using the KEGG reaction to understand which metabolite should be produced. In this example, the reaction R01082 produces C00149. Since C00149 is synonymous with mal__L, the FUM reaction must be forward or bidirectional to produce mal__L, and be considered synonymous with R01082. There may be cases when this strategy fails to find synonyms for a given reaction in the model. When this happens, a second strategy is used. In this example, the first strategy fails because, even if 3 metabolites are present in both reactions, hydrogen is only present in the big reaction, and the metabolite R5P is not described as a synonym of C00117. However, these two metabolites are very similar, differing only by 2 hydrogen atoms. In these cases, we use the information of synonymous reactions, and RBL is indeed described by MNXRef as a synonym of R01051, even though not all metabolites are synonyms. A third strategy, less strict, is used for the reactions that still do not have a synonym identified. For example, the first strategy fails to associate ADK3 to R00430 because 2 of the metabolites in the ADK3 don't have a synonym in R00430. The second strategy fails because MNXRef does not link ADK3 to any KEG reaction. However, the 2 metabolites present in both reactions are exactly the ones used as substrate and product in the guanine biosynthesis module. This means that, in this specific KEG module, ADK3 can act as R00430. In this third strategy, if ADK3 can replace R00430 in every KEG module, ADK3 is mapped to R00430. This method is used to map models' reactions to KEG, but not to biopsych. The model IAF692 illustrates the importance of each of the described mapping methods. Using the first method, 66% of the metabolic reactions were mapped to KEG. The second and third methods only contribute to an extra 4% and 3% of the mapped metabolic reactions. 27% of the metabolic reactions cannot be mapped to KEG. Returning to the analysis report, in the tables subsection, you can find the number of processing steps of each pathway identified in the model. The KEG modules are organized into categories, such as carbohydrate metabolism and energy metabolism. You will also notice that the table has a color coding. The green cells in the table correspond to modules that are complete in the model. The yellow cells have a completeness level lower than 100% and higher than 0%. When you place your mouse pointer over the number of processing steps, you can see the KEG IDs of the reactions present in the model, as well as the number of reactions. Note that the number of reactions can be different from the number of processing steps. To clarify the difference between reactions and processing steps, we will click on the completeness link, which will open a KEG map of the selected pathway. The KEG map highlights in red the metabolites and the reactions present in the model. The ellipses represent metabolites, and the rectangles represent reactions. The consumption of the metabolite C00158, for the production of C00311, is one processing step, but it can be done either by one reaction, or by 2 reactions. When a processing step needs 2 reactions, it is not possible to highlight it in red, but this information is present in the table. These 2 reactions on a single processing step explain the difference between the number of processing steps and reactions. Besides the completeness, the number of blocked reactions and essential reactions is also shown for every module. Like in the completeness level, it is also possible to visualize the essential reactions in a KEG map. Essential reactions can only be identified if the model has a predefined objective reaction. For this output, it was not selected the name of the modeled organism. Thus, a possible strategy to find the pathways present in the model is to list all the pathways that use at least one reaction present in the model. For example, by clicking on the pentose phosphate pathway, and then the first reaction present in the model, we find a list of all modules using this reaction. If we apply this procedure for every reaction present in the model, the number of pathways listed would be very large, and most of them would have a low completeness value. Therefore, SbmlComp uses MinPath to find the smallest number of pathways that together use all the reactions mapped to KEG. A practical outcome of this decision is that the pentose phosphate cycle is not listed as one of the present pathways, as its presence is not necessary to explain the reactions present in the model. Another possibility for finding out the pathways present in the model is by using the organism's annotation made by KEG and biopsych. To explore this second possibility, we will change to another analysis report, which used as input the same model as in the previous analysis, but Escherichia coli was selected as the modeled organism. Scrolling down towards the KEGG tables, you will notice that this analysis shows many more pathways than in the previous example. This is because all pathways annotated by KEGG for this specific organism are present in the table. Because the pathways have been annotated, you can find in the table the number of annotated processing steps present in the model. For example, the citrate cycle has 6 annotated processing steps present in the model. Annotated usually registers the same number of processing steps as entire module. However, when the entire pathway was not annotated on the organism, the two categories will have different completeness values. For example, the glycolysis core pathway has a 100% completeness for annotated. By clicking on the completeness link, we can see in the KEGMAU that the reaction R07159 is not annotated in the organism. For this reason, the completeness is 86% for the entire module. If the reaction R07159 were present in the model, there would be 100% completeness in the entire module, but the pathway would also have an unpredicted processing step. The category unpredicted registers the number of not annotated processing steps present in the model for a given pathway. The category annotated missing registers the number of annotated processing steps missing in the model. For example, all yellow cells should have at least one missing processing step because they have a lower than 100% completeness. Pink cells are only present when the organism name is indicated, and it is reserved for annotated pathways with a completeness value equal to 0%. Blue cells are only present when the name of the organism is selected, and the model has reactions not present in any annotated pathway. To map these reactions to a pathway, MinPath is used. Because blue cells are reserved to pathways not annotated on the selected organism, they do not have the categories annotated or annotated missing. This means that every blue cell will have at least one unpredicted processing step, which is not present on any annotated pathway. For example, the methylaspartate cycle module, on the E. coli core model, has 5 processing steps in the entire module. Only one is listed as unpredicted because the other 4 are present on other pathways. An interesting aspect to check is whether a pathway is functional or not. This might be especially important for pathways predicted by MinPath, as there is no experimental data to support the existence of the predicted pathways. However, it is possible that a pathway has gaps and it is still functional. For example, the TCA cycle has 60% completeness. By clicking on the completeness link, you can see that 2 reactions are apparently missing in the model, R01197 and R02164. However, the metabolites used as substrate and product on these 2 reactions are present in the model, and multiple reactions may be working together to take over the role of the missing reaction, making the pathway functional. Scrolling down again, in the next subsection, you find some of the results from the table visualized in a heat map. However, only pathways with 4 or more processing steps, and at least 50% completeness, are represented in the heat map. The first data in the heat map is the pathway completeness. In a second column is the percentage of essential reactions in the pathway. The number of model reactions present in each pathway is also shown. This allows for a perception of the pathway size, as the 2 previous columns are percentages and do not carry information about the actual size of the pathway. Finally, it is present in the heat map the minimum and maximum total flux needed in each pathway to obtain the maximal growth rate. To explain better the concept, let us use an example. Let us say that you want to produce the red metabolite using the blue metabolite as substrate. This example includes 2 possible routes, through pathway A and pathway B. When the route through pathway A is chosen, because this reaction uses one metabolite as substrate and produces 2 metabolites as products, there is a total production of 2 red metabolites. On the other hand, on pathway B, one substrate generates 1 product, and therefore only 1 red metabolite is produced. In this scenario, there are no alternative routes that allow maximum production of the red metabolite, and no flux should go through pathway B. This also means that maximum and minimum flux in pathway A will be equal. However, when changing the network to produce the same amount of the red metabolite using pathway A or B, there is an alternative route to obtain maximum production of the red metabolite. Therefore, To obtain the maximum production of the red metabolite, there can be maximum flux in pathway A or pathway B. The minimum flux in both pathways is 0 because the flux can be redirected to another pathway, and the red metabolite can still have maximum production. Back to the analysis report, the last two columns of the heat map show the same kind of analysis of the example, but it is a sum of the minimum and maximum flux of each reaction participating in the pathway. The FVA function of Cobra Pi is used for this analysis. It is interesting to compare, side by side, the heat maps obtained when the modeled organism is informed and when it is not. In the first case, the primary source of pathways present in the model is the organism annotation, and in the second case, the pathways are suggested by MinPath. Without annotation information, the percentage is calculated for the complete pathway, which might lead to a lower completeness value. You can notice a difference in the completeness level in the glycolysis core module, which is 85.7% for the entire pathway, but 100% for the annotated pathway. You can also notice that 2 extra modules are present in the heat map when using annotation data. The Emden glycolysis module is one of these extra modules present only in the annotation data. To explain the reason for this, let us open, side by side, the KEGG maps of the glycolysis core module and the Emden pathway. In the Emden pathway, the first reaction present in the model is R01015. This reaction is also present in the glycolysis core, and both modules are identical from C00111 downwards. Because there are no reactions in the model exclusive to the Emden pathway, and because MinPath tries to minimize the number of pathways, the Emden pathway will not be predicted and therefore is not present in the heat map. This is the same reason why the pentose phosphate pathway was not predicted by MinPath, despite being annotated. These differences aside, it can be said that MinPath does an excellent job at predicting pathways present in the organism, at least for pathways with more than 50% completeness and at least 4 processing steps. Scrolling down in the web page, you will find a similar table as the KEGG pathway modules, but for biopsych pathways. Just like in the KEGG table, you can click on the completeness value, and be forward to the pathway map. However, it is not possible to highlight the reactions present in the model. Also, the number of conversion steps is always equivalent to the number of reactions. Finally, biopsych pathways are usually in higher quantity than KEGG pathways modules, as KEGG has 300 modules, while biopsych has 2,936 pathways. You can also find a heat map similar to the one created for the KEGG results. Scrolling down again, you find growth simulations of the model on multiple sources of carbon, nitrogen, phosphorus, and sulfur, as well as negative controls to check if the model can grow without carbon, nitrogen, phosphorus, or sulfur sources. The growth media used in the simulations is inspired by the biolog phenotype microarray experiments, and the simulation results are displayed in a figure similar to the biolog microarray. Plates 1 and 2 are used to test 190 carbon sources. Plate 3 is used for nitrogen sources. The plate 4 is used to test phosphorus and sulfur sources. This analysis is only carried out on models with a defined objective reaction, and you can check the name of the model and the identifier of the objective reaction in the first 2 lines of this section. To better understand how the biolog experiments work, we can use the carbon source experiments as an example. The negative carbon control has nitrogen, phosphorus, and sulfur sources in its composition, but has no carbon sources. Every other simulation with carbon sources has the same metabolites present in the negative control, plus a single carbon source. By clicking on the carbon negative control and in the L-arabinose, you can find the detailed media composition. By putting both media compositions side by side, we can see that the 2 media differ only by the L-arabinose. This information is provided by biopsych and will help us to understand the color coding used to display the simulation results. First, you have yellow circles, which indicate that the model cannot grow in the medium. In this example, the model was not able to grow without a carbon source. A second color is brown, which indicates that the model does not have any reactions capable of using that particular carbon source as a substrate. Because the culture media differs from the negative control by a single carbon source, the media becomes identical to the negative control when this molecule cannot be metabolized. The color pink indicates the media where the model can grow. When the mouse pointer is placed over the culture media, the optimization result is shown in a floating window, together with the experimental growth phenotype of E. coli. You can check that the model has a higher growth rate at some media than in others. This particular model is a small E. coli model, with little variety of metabolites. This is why most of the growth media are brown. Interestingly, this model can grow on the sulfur negative control, which indicates that this model does not represent sulfur-related pathways well. In the tables above the illustration, you find a summary of the model's growth phenotype. In particular, you find the phenotype accuracy if the model represents an E. coli. It is also interesting to note that this specific model had a high accuracy in sulfur sources because it can grow without a sulfur source, and thus can grow in every sulfur source. This indicates that models can have high phenotype accuracy, even with fundamental errors. To better understand how the growth simulations run, we will change to another analysis. This new analysis report compares 3 models, all of Escherichia coli. Going back again to the growth phenotype subsection, and then to the analysis of IAF 1260, we can find more information about the growth media. SbmlComp tries to obtain model growth in at least one medium while using the default biolog media, but this is not possible in many cases, and thus it is necessary to include extra metabolites in the growth media. One reason for changing the default medium is because some models have biomass reactions that use as substrate metabolites that are not produced internally by the model, but are obtained directly from the extracellular medium. Therefore, for the model to grow, it is indispensable that these metabolites are present in the culture medium. You can find these metabolites listed as extra metabolites, present in the objective reaction, included on every external media. To determine which metabolites are necessary to be included, SbmlComp searches for the minimum set of medium metabolites defined in the model file that need to be kept for the model to grow in at least one media, without growing on any negative controls. This means that the extra set of metabolites are obtained from the culture medium of the model file itself. In fact, this minimum set identification is performed individually for the carbon, nitrogen, phosphorus, and sulfur sources. The metabolites present in the 4 solutions and in the objective reaction are placed in the first category. The metabolites present in the 4 solutions, but not in the objective reaction, are included in the list extra metabolites, not present in the objective reaction, included on every external media. If you ask for oxygen to be included in the growth simulations, and the model can metabolize oxygen, this is where oxygen will be listed. The metabolites that are not present in all 4 solutions are listed in individual categories, like extra metabolites included in the external media when testing carbon sources. Similar categories exist for nitrogen, phosphorus, and sulfur. This is the end of the model analysis section. Going down to the pairwise model comparison section, we can see that 3 models are present in this analysis report, and that every model is compared against every other model. Clicking on the subsection E. coli core and IAF 1260, you find 3 Venn diagram. The first diagram shows the overlapping of exchange reactions and sink reactions between the 2 models. Exchange and sink reactions were aggregated because they have a similar role in the model. All 7 exchange reactions in E. coli core are also present in IAF 1260. This means that E. coli core shares 100% of its media with IAF 1260, and that IAF 1260 shares 31.8% of its media with E. coli core. Below, you will find a second Venn diagram, where you can identify the number of metabolic reactions exclusive to one of the models, and shared between the 2 models. Checking the table used as a reference to plot the Venn diagram, you will notice that the number of E. coli core reactions in IAF 1260 is not the same as IAF 1260 in E. coli core. This might seem strange, and therefore it is helpful to explain this result in more detail. E. coli core has 50 metabolic reactions, and 2 of them are not present in IAF 1260, the biomass reaction and the FRD7 reaction. On the other hand, IAF 1260 has 1,355 metabolic reactions, and 50 of them are present in E. coli core. To understand this, we need to know that GLUN and GLUNPP are among the 50 reactions of IAF 1260 mapped to E. coli core, and that both of them were mapped to the same reaction. GLUN and GLUNPP are actually the same reaction, but in different compartments. Because SbmlComp disregards compartments during mapping, both reactions are mapped to the same E. coli core reaction. The ATPM and NTP1 reactions, present in IAF 1260, are both mapped to the same reaction in E. coli core because they are essentially the same reaction. Back to the analysis report, the last Venn diagram shows the number of biologic media where only E. coli core is able to grow, where only IAF 1260 is able to grow, and where both models have the same growth phenotype, either by growing or not growing. The sum of the three areas in the diagram equals 383, which is the total number of culture media on the 4 biologic plates. Below the Venn diagrams, you find a bar graph for the modules/pathways comparison between the two models, and you can find the KEGG pathways modules identified in both models or in only one of the models. The color coding on this chart is similar to the ones used on the Venn diagram: yellow for the pathways present on both models, red for the ones exclusive to E. coli core, and green for the one exclusive to IAF 1260. Only pathways with at least 4 reactions, and with a minimum of 30% completeness, are shown in this graph. Some pathways are equally present in both models, while other pathways have higher completeness on IAF 1260. In this example, no pathway, or part of a pathway, is exclusive to E. coli core. Possible reasons for this are: the 2 reactions that are exclusive to E. coli core do not participate on any pathway, or are among the 12 reactions of E. coli core that have not been mapped to any KEGG reaction, or are present in a pathway shorter than 4 reactions, or with less than 30% completeness. Note that the KEGG pathway modules in this bar chart are organized into categories, such as carbohydrate metabolism and amino acid metabolism. The biopsych bar chart follows the same color code as the KEGG chart. One relevant difference is that the pathways are organized into 2 categories: pathways and superpathways. The superpathways are conglomerates of smaller pathways. Another section that is only visible when more than one model is used as input is the model's distance, where the distance between the models is calculated and represented in a metric multidimensional graph, and in a hierarchical clustering dendrogram. The input data to create these graphs is a distance matrix populated with the jagged distance coefficient of each model pair. The distances between models are calculated for metabolic reactions, exchange reactions, and growth phenotype. These are the same comparisons made on the Venn diagrams. The last section of the analysis is the model export, where you can modify the model by including or excluding reactions of a specific pathway. To select the model you want to modify, you just have to click on the model's name. The first way to modify the model is by including in the model reactions annotated by KEGG and biopsych for the selected organism. Using this option, you can complete the pathway using the reactions annotated by the databases. This is done by translating the annotated reactions into the nomenclature system of the model. However, some reactions cannot be translated. For example, if you have a model that uses BigIDs, and you want to include in your model all the methanogenesis pathways from KEGG, some reactions will not be included, as BIG has no IDs for some of the reactions present in these pathways. If you wish to select all the pathways, you can click on Select All. Below, you have the option to remove from the model reactions annotated by KEGG and biopsych on the selected organism. This option can be useful if you want to simulate the phenotypic change of the model when a pathway is knocked out from the model. Below, you find the option to remove from the model reactions mapped against KEGG and biopsych pathways annotated on the selected organism. This option can be useful to identify possible reactions erroneously included in the model. However, reactions will only be removed from the model if they are exclusive to the selected pathways. Since the same reaction can be present in multiple pathways, this will avoid changing unselected pathways. Below, you have the option to remove from the model reactions mapped to KEGG and biopsych pathway not annotated on the selected organism, and that cannot contribute to maximum growth. This option searches for the intersection between the previous option, and the pathways without flux listed in the minimum total flux heat map. This option can be used to remove possible unwanted reactions, but more conservatively than with the last option. Finally, there is the option Include Reactions from Another Model. This option is only visible when more than one model was included as input in the tool, and it is useful if you have a reference model and want another model to have the same metabolism capabilities for a given set of pathways. When including reactions, new metabolites might be included in the model. One way to prevent these new reactions from being blocked is by including demand reactions, as they remove from the system metabolites not consumed by other reactions. SbmlComp will also try to unblock all reactions listed as blocked in the model analysis section when this option is selected. When the Build Model button is clicked, the changes to the model will be processed, and a zip file will be available for download. Inside this zip file, you will find the modified version of the model, and a TSV file with the changes made to the model. For now, this is all. Have fun using the tool.